Good morning, everyone. My name is Raquel del Campo, and today I will be talking about the topic that my thesis will follow, that is viruses as biocontrol agents of the alder decline. The alder decline is a widespread disease that affects mainly all alder species. The main symptoms reflect a progressive decay of the tree that is mainly visualized by the cankers on the trunks, the death of some apical branches, also root death, and it could also have adventitious sprouts. It is known that this disease is produced by homicides belonging to the Phytophthora alni complex. The Phytophthora alni complex is formed by 3 different species, alni, multiformis, and uniformis. But however, more species have been isolated from disease alder trees. So we don't know exactly how this disease is starting or which organism is the cause of this disease. The Phytophthora species from the homicide gender are more similar to filamentous fungi in habitat and morphology, but they are more phylogenetically related to the atoms and brown algae. The main difference between the real fungi and the homicides is that the asexual reproductions occurs through zoospores that can move, and also the cell walls are made by cellulose instead of chitin, like the real fungi. It's also interesting to mention that all the Phytophthora species that are known are very well-known aggressive pathogens. And so, like other organisms in the planet, these Phytophthora species that are involved in the alder disease will have mycoviruses in their genomes. Mycoviruses were first discovered in 1948 in a disease that was affecting Agaricus bisporus. But the most important highlight in the mycovirus history was in 1975 when the Cryphonectria parasitica hypovirulent 1 was discovered. The mycoviruses affecting fungi and homicides are characterized by a persistent intracellular non-lytic lifestyle, and also by the transmission via spores and aphal contacts. Natural cross-kinder transfer has also been discovered in these mycoviruses. Mycoviruses are fully persistent in all the fungal kingdom, and they are classified in 23 families and a genus that is not well classified yet. They are classified depending on the genome type, and almost all fungal and homicide viruses have this double-stranded RNA and single-stranded RNA, but there are many funguses that also can have single-stranded DNA. The most important sequence and more conservating sequence in this fungi is the one that encodes the protein RNase-dependent RNase polymerase, that is shortened as RDRP. And these mycoviruses can cause effects on the host by producing hypovirulence or non-effect, or they could be hypervirulent. When we talk about hypovirulence, some fungi can cause effects like abnormal pigmentation or irregular growth, or maybe problems in the sporulation. As for example, this fungi that I'm presenting here, that is the fungi that is the virus, that I mentioned before, the Cryphonectria parasitica hypovirulent 1, that if you see the picture, what you see is that this virus is affecting the color pigmentation of the fungi, and is also affecting the sporulation. When we talk about hypervirulence, what comes to my mind are 2 different effects. It can affect plants, conferring resistance to them, like Leptosphaera biglobosa quadrivirus does, that is not conferring the resistance to the plant that affects this Leptosphaera biglobosa, but it's conferring resistance to another fungi that is more aggressive. Another use of these hypovirulent viruses are the effects in the entomopathogenic viruses that are very used to control some pests, some arthropod pests. And this is mainly affecting the fungi from the genera that are showed here in the slides. But the problem is that not many viruses can cause these effects. So using all this information that we have obtained from the mycoviruses research, we will apply it to more technologies. For example, a technology that is applied is the interference RNA. The interference RNA is a plant and fungi defense system, again, double-string RNA. That is basically an antiviral system that ends in a post-transcriptional gene silencing. I'm going to explain a bit the system, but basically the cell, in the cell there is a protein that is called dicer that recognizes this double-string RNA and cleaves it. The cleaved RNA are recognized by a RISC complex, and this RISC complex finds complementary mRNAs that will finally be cleaved as the other ones. The cleave of these mRNAs leads into the gene silencing. Two main techniques have been developed with this system, involving this system. First, the SIGS, which Uma have mentioned before, that it means spray-induced gene silencing, and also another technique is the VIGS, that is vector-induced gene silencing. I will be focused more on this technique. The main difference between these two techniques is the entrance of the dsRNA and the persistence of these molecules in the cell. Viral-induced gene silencing in plants have been applied successfully. For example, in this study, they applied this technology using a vector that was constructed with a plant viruses and some genes that were involved in the infection process. In this study that you can see here, they found that some genes that were called RXLL were silenced in plants, leading to the resistance of the plant to the virus. But this technique, what I want to do is apply these techniques to fungi, and this is not still good developed. There's only one study that have demonstrated that mycoviruses can be used as VIGS. So this is a good thing to consider, and I will be focused more on this during my research. So all this information made me question things like, could viruses infecting Phytophthora species involved in alder dieback have any effect? Is it possible to apply VIGS technologies with mycoviruses in Phytophthora? Well, I still cannot answer these questions because I have just started my PhD. But what I can mention to you is what my objectives will be. First will be to study the whole biome of all the Phytophthora species that cause the alder dieback. Second, I would love to know the effect of those viruses cause. And the third one, I will like to develop the VIGS tool for the Phytophthora species in order to produce hypovirulence trait. Hopefully in the next Congress I will be able to answer those questions, but for now, if you have any question about mycoviruses or the applications in the biocontrol agents, I will be here to answer whatever you want. So thank you for your attendance, and that's it.